Usually, when we talk about a medical diagnosis, there is this expectation of precision.
Right, like it's black and white.
Exactly. It feels like engineering. You break your arm, the X-ray shows that jagged white line, and the doctor just points to the screen and says, there it is, broken.
It's very binary.
Yeah. Broken or not broken, it's clean, it's visible, and honestly, it's deeply comforting because it can be categorized so easily. But then you step into the world of neurodevelopment.
Oh, yeah. That's a whole different ballgame.
And rare genetics, right.
Yeah.
And suddenly that X-ray machine is just useless. You aren't looking at a clean break anymore. You're looking at a diagnostic landscape that is entirely murky.
It is the absolute definition of diagnostic muddy waters, I mean.
Muddy waters is a great way to put it.
Yeah, because you are searching for a typo in a biological manuscript that is, 3 billion letters long?
3 billion. It's just unfathomable. So if you're listening to this, you are likely a parent or, a carer, a grandparent, maybe a sibling of a child with PURA syndrome.
Mm-hmm.
And today is a massive milestone for us.
It really is.
This is day 31. This is the final day of our 31-day deep dive into PURA syndrome. And over the last month, you have been in the trenches with us.
Deep in the weeds.
Right. You've spent weeks absorbing the clinical reality, all that overwhelming medical jargon, the daily management strategy.
The neurology, the gastroenterology.
All of it. But today is different. We are capping off this intense month of learning by synthesizing a stack of incredibly forward-looking sources.
Which is so refreshing after all the heavy medical stuff.
It is. We've got the PURAcure Roadmap, which was commissioned by Jax tomorrow. We have updates straight from the PURA Syndrome Foundation.
And the Natural History Protocol from the University of Southampton.
Yes, and updates from the Jackson Laboratory. And even these deeply personal parent workbooks from experts like Sharu Azadi. We are basically looking right at the horizon.
Yeah, we're pulling all of these threads together to map out the incredible momentum that is happening right now in the PURA community.
I mean right now.
Right now, not 10 years from now. From groundbreaking genetic models being bred in labs to, new therapeutic pipelines to the global family support network that is advocating for your children every single day.
So this deep dive is your shortcut to understanding the hopeful future that's being actively, aggressively built for your child.
I love that word. Aggressively built.
It's true. So as you were listening to this, maybe you're driving to a therapy appointment, or maybe you're just making breakfast or sitting in the quiet of the early morning. I want you to remember this. You spent 30 days in the weeds. Today, we focus on the progress.
The community and the profound hope on the horizon.
Exactly. And, there are cars in my driveway that are older than the medical diagnosis of this condition.
That is a wild way to think about it.
But it's true. We have to start by acknowledging just how remarkably short the history of PURA syndrome actually is.
Yeah, the very first clinical paper to recognize PURA syndrome as a distinct genetic condition was published on October 23, 2014.
2014?
Right, by a team led by Dr. David Hunt at the University of Southampton. Before that date, this syndrome did not exist in the medical literature.
He had no name.
No name at all.
That timeline is just staggering to me. In 2014, when it was first discovered, there were just, 15 reported patients?
Yeah, just 15 in those initial studies.
15 families in the entire world who finally had a name for what their child was experiencing. And today, the landscape looks completely different, doesn't it?
Completely. We are currently looking at an estimated 706 to 840 diagnosed cases globally.
Wow. And those families are spread across 60 different countries, right?
Yeah. Argentina, Japan, Australia, Denmark, the UK, the US, India, everywhere.
And because modern genomic testing like whole exome and whole genome sequencing is becoming so much cheaper and, much more widely accessible, the expectation is that hundreds more will be diagnosed in the next few years.
Absolutely. And understanding those new diagnoses really requires looking at the nature of the mutation itself.
Right. Let's get into that.
So in the vast majority of cases, PURA syndrome is caused by a de novo mutation.
De novo.
Right. De novo is a Latin term. It basically means of new. In genetics, it means the mutation happens spontaneously.
Spontaneously, like just out of nowhere.
Yeah, during the formation of the egg or sperm, or very, very early in embryonic development.
So it's not inherited.
No.
It wasn't lurking in your family's DNA for generations. It started completely fresh with your child.
Exactly. And to understand the implications of that, we have to look at what the PURA gene actually does in the body.
Okay, lay it on us.
Think of every gene as essentially an instruction manual for building a specific protein.
Okay, instruction manual. I like that.
The PURA gene provides the instructions for making a protein called Pura Alpha. Now, Pura Alpha is a multifunctional protein, but its most critical roles involve regulating how DNA replicates and how mRNA is transported within the cell.
So it's busy. It's doing a lot.
Very busy. It is absolutely vital for normal brain development. And every person has 2 copies of the PURA gene, one inherited from each parent.
Right, standard genetics.
But in PURA syndrome, one of those 2 copies gets altered or deleted.
And because it is de novo, I just love this analogy I've seen floating around, the chosen family tree.
Oh, that's a beautiful way to phrase it.
Because, you don't inherit the mutated gene from your ancestors. You didn't get it from your great-grandmother. But the second you receive this diagnosis through organizations like the PURA Syndrome Foundation and PURA Foundation Australia, you instantly inherit a global family tree of support.
Yes, you are immediately grafted onto this incredibly robust, worldwide network of people who understand exactly what your daily life looks like.
And that connection is critical for survival. I mean, not just emotional survival, but literal, practical survival.
Oh, without a doubt.
Think about those families before 2014. They were wandering in the dark. Their children had severe neurodevelopmental delay, profound hypotonia, which is that low muscle tone, the floppiness.
Seizures, feeding difficulties.
Right. But without a specific genetic target, a lot of these children were misdiagnosed.
Yeah, they might have been told they had Rett syndrome or myotonic dystrophy or central hypoventilation syndrome.
The parents were essentially fighting a ghost.
Yeah.
But now, the moment a family gets those whole exome sequencing results back, they are plugged into this hive mind of shared experiences.
Which is so necessary because the isolation of a rare disease can be as debilitating as the physical symptoms themselves.
I can only imagine.
And this ecosystem of support, it extends far beyond just the primary caregivers. The Foundation updates highlight a group called Grand POPS.
Grand POPS, I love that name.
It's the grandparents of PURA syndrome. They are actually holding virtual meetups, with one scheduled for February 28th, 2026.
Recognizing that brothers and sisters and grandparents are also on this rare disease journey is just brilliant.
It really is. Grandparents experience a very unique, almost dual-layered type of grief and joy.
How so?
Well, they are watching their own child navigate the immense, exhausting burden of being a rare disease parent, right? While simultaneously navigating their own relationship with a grandchild who requires complex medical care.
That is a lot to process.
And grandparents often have a bit more time to advocate or assist, but they lack dedicated spaces to process those complex emotions without feeling like they are burdening the primary parents.
Yeah, because they don't want to complain to the parents who are in the thick of it.
Exactly. And the same applies to siblings. Siblings might struggle with the fact that their brother or sister requires a disproportionate amount of their parents' attention and energy.
Right, which is completely natural.
Bringing siblings together at conferences or online meetups, it normalizes their daily reality. It provides a mirror. It tells them, your family structure is not an anomaly. There are other kids living the exact same dynamic.
Here's where I want to push back just a little bit, though.
Okay, go for it.
Emotional support is beautiful. It's wonderful that a grandparent in the UK can log on to Zoom and talk to a grandparent in Australia. And it is vital that a mom can ask a Facebook group, hey, what AAC device works for your kid?
Yeah.
But how does a Facebook group or a grandparent meetup actually move the needle on treating the syndrome? I mean, emotional support doesn't sequence DNA. How does community actually lead to a cure?
That is a great question. The reality of modern biomedical research is that a highly organized, connected patient community is the absolute non-negotiable prerequisite for attracting researchers and funding.
Wait, so the community comes before the science?
In ultra-rare diseases, yes, absolutely.
Really?
Emotional connection builds the foundation for trust, and trust builds the foundation for data collection.
Okay, break that down for me.
Imagine you are a brilliant geneticist at a major university. You have the funding and the technology to study a rare gene. But to do that, you need patience. You need comprehensive medical data. You need biological samples. If the 800 PURA patients in the world are scattered, disconnected, and isolated, your research will stall before it even begins because you simply cannot find the subjects to study. But if you have a foundation that has already organized those families, already earned their deep trust, and can send out a single email saying, hey, we have partnered with this researcher and we need 50 families to participate, Well, the science can move at lightning speed.
So the Facebook group is literally a mobilization tool.
Exactly. The community is the engine that drives the science.
That makes perfect sense. The community is the infrastructure, and that infrastructure is currently being mobilized to build registries and biobanks,
Yes.
like the Global Patient Registry, which is being run in collaboration with the clinical informatics team at the University of Southampton. Now, it's described in the sources as a longitudinal natural history study. What exactly does that mean?
So, natural history study is a foundational phrase in rare disease research. A natural history study tracks how a disease progresses naturally over time, over a patient's entire lifespan without any therapeutic intervention.
Okay, so just watching what happens.
Right. It meticulously tracks developmental milestones, neurological history, the frequency and type of seizures, the progression of orthopedic issues like scoliosis or hip dysplasia. It is the holy grail for scientists because it creates a baseline.
But doesn't the medical community already know what the disease does? I mean, they diagnosed it.
Not with a disease discovered in 2014. We do not have decades of textbook data outlining the trajectory of a PURA patient from infancy to late adulthood.
Oh, right. The adults with PRA now weren't diagnosed when they were kids.
Exactly. And more importantly, to know if a new experimental drug actually works, you must first know exactly how the disease would have progressed without the drug.
Okay, let me see if I have this right. So if you give a 5-year-old with PURA a new experimental gene therapy, and at age 7 they start vocalizing new sounds, and their seizure frequency drops by half,
Right?
you need to know if that was the drug, or if kids with PRA just naturally develop new sounds and have fewer seizures at age 7 anyway.
Precisely. You can only prove the drug's efficacy if you have a massive data set, a natural history baseline that definitively states, without intervention, only 2% of PURA patients vocalize new sounds at age 7, and seizure frequency typically increases, not decreases.
Got it.
The registry creates the empirical ruler against which all future cures will be measured. Without the natural history study, the FDA will look at your clinical trial data and say, we have no proof your drug did this. Maybe it was just time.
So you, the listener, filling out an annual online questionnaire about your child's sleep patterns or feeding tube usage or physical therapy progress, you are literally drawing the map that scientists are going to use to prove a cure works.
Yes, your late-night data entry is a critical piece of the scientific method.
That is so empowering. But it goes beyond data entry too, right? There is the global biobank.
Yes, the PURA Syndrome Foundation has partnered to create biorepositories in the US, Germany, and Australia. These biobanks are collecting biological samples, specifically skin biopsy cells, fibroblasts, and blood from globally consented PURA patients.
Because a researcher in a lab in California doesn't have the time or money to fly to Denmark to draw blood from a patient.
Exactly. They just request a frozen sample of living cells from the biobank.
It democratizes the research.
It does. But the bridge between the patient's lived experience and the clinical lab requires more than just cells. It requires a shared language.
A shared language.
Yes, and this is where the disease concept model, or DCM, comes in. The foundation is developing this in collaboration with an organization called Combined Brain.
Okay, disease concept model. That sounds incredibly dry and academic.
It sounds academic, but it is deeply, deeply personal. It is a structured framework that aligns the underlying microscopic biology of PURA with the macroscopic symptoms that actually matter most to the patients and their families.
Give me an example of that. Because a scientist looking through a microscope might say, what the most important thing to fix is restoring the exact cellular concentration of the pure alpha protein in the dendrites of the neuron.
Right, that's the scientist's view. But a parent says, I don't care about the dendrites. The most important thing is that my child stops having drug-resistant seizures so they can sleep through the night, and that they have enough core strength to sit up and use their communication device.
So the scientist and the parent are looking at the exact same problem, but from completely different altitudes.
Exactly. The disease concept model translates those lived experiences, the daily seizure tracking, the severe fatigue, the hypotonia, into clinically meaningful endpoints.
Oh, so it translates parent goals into FDA goals.
You nailed it. It ensures that when pharmaceutical companies design clinical trials and when regulatory bodies review those trials, they are measuring success using the same metrics that the parents use. It forces the science to serve the practical needs of the patient.
That is brilliant. But I mean, with a syndrome this rare, let's say 800 people globally, is there really enough data to get big pharmaceutical companies to pay attention?
It's a fair question.
Because 800 people worldwide is a tiny, tiny market for a drug company that needs to recoup, billions of dollars in research and development.
Historically, yes, that was a massive barrier. But we are entering an era of precision medicine. We aren't just throwing broad-spectrum chemicals at symptoms anymore. We are targeting the exact broken line of genetic code.
So the rarity isn't as much of a deal breaker.
Not anymore. While the data pool for PURA is small, the precision of the genetic target makes it highly valuable. When you combine robust patient data with living, breathing models of the syndrome in the laboratory, it creates a highly attractive, de-risked pipeline for researchers and biotech firms.
So if the mouse is the testing ground, what are we actually injecting into it? This is where the roadmap moves from observing the broken gene to actively rewriting it.
Yes. Let's look at the basic biology and the living models.
Right. JAX Tomorrow, which is a nonprofit focused specifically on funding treatments for PURA, commissioned the PURAcure Roadmap, authored by Dr. Ethan Perlstein and his team.
The roadmap is essentially a master plan. It identifies the critical gaps in our basic scientific knowledge and lays out a prioritized, step-by-step path to finding therapeutics.
Okay, so what's step one?
Well, one of the biggest gaps they identified is that we still don't fully understand the mechanism of the disease. We know that the PURA gene is mutated, but we don't always know how those different mutations actually break the cellular machinery.
Because there are over 90 different pathogenic variants reported. Let's break down what that means, because mutation is a really broad term.
Yes.
Some are deletions, where a literal chunk of the DNA code is just missing. Some are nonsense mutations. And a nonsense mutation is like a period placed in the middle of a sentence. It tells the cellular machinery to stop reading the instructions and stop making the protein way too early, resulting in an abnormally short, usually non-functional protein.
Exactly.
And then there are missense mutations. That is where a single letter in the genetic code is swapped out for the wrong one. So one single building block of the protein is incorrect.
And the roadmap points out that we need to know exactly how these different variants affect the Pura Alpha protein's ability to do its job. We need to determine if the disease is caused by haploinsufficiency or if there is a dominant negative effect.
Okay, let's slow down here. Haploinsufficiency and dominant negative. These are massive, complex biological concepts, but they basically determine the entire drug pipeline.
They absolutely do.
Let's use the factory analogy.
I love the factory.
Okay, so every cell is a factory. You have two assembly lines, your two alleles, one from each parent, and both are producing pure alpha protein. Haploinsufficiency means one of those assembly lines breaks down completely. It produces nothing. The other assembly line is working perfectly, operating at 100% capacity. But the factory as a whole is now only producing 50% of the normal amount of pure alpha.
That's right.
And for some genes, 50% is plenty. The factory survives. But for the PURA gene, 50% is just not enough. The cell is insufficient with just one haploid or single working copy.
That is a perfect explanation of haploinsufficiency. You simply do not have enough of the required material. But a dominant negative effect is entirely different and much more insidious.
Okay, how does that look in our factory?
In a dominant negative scenario, one assembly line is working perfectly, making good, healthy, pure alpha protein.
Okay.
But the mutated assembly line isn't just broken and idle. It is actively producing a warped, malformed, toxic version of the protein.
Oh, wow. So it's making garbage.
Worse than garbage, because proteins often bind together to do their jobs. This rogue, toxic protein latches onto the healthy proteins produced by the good assembly line and drags them down, destroying their function too.
Oh, that is insidious.
Right. The mutated copy is dominant and has a negative effect on the healthy copy. It is an active saboteur, not just a passive absence.
That distinction is huge, because how you fix a factory that is missing half its output is completely different from how you fix a factory that has an active saboteur on the floor.
Exactly. And to figure out which scenario is happening with each of the 90 variants, you need living models.
And in May 2025, there was a massive breakthrough on this front.
There was. The Jackson Laboratory, commonly known as JAX, which is an absolute powerhouse in biomedical research, along with JAX Tomorrow, announced they had successfully created a new knockout mouse model for PURA syndrome.
This was a pivotal moment.
It was. The JAX team used CRISPR-Cas9 to create this model.
CRISPR, which we've talked about before.
Right. CRISPR is essentially a highly programmable pair of molecular scissors. Scientists create a guide RNA that matches the exact sequence of the PRI gene they want to target. The Cas9 enzyme follows that guide, finds the exact spot on the DNA strand, and cuts it.
Just snips right out.
Using this technology, they cut out a portion of one of the 2 copies of the PRA gene in mice, intentionally replicating the genetic condition of a human with PURA syndrome. They call it the PURA-co mouse.
And what happens when you knock out that gene in a mouse?
They exhibited what scientists call a strong phenotype. Phenotype is basically the observable physical and neurological traits.
Right.
And the symptoms were very clear and very consistent. These knockout mice produced significantly less PRI protein, they experienced fatal seizures, and they had significantly shorter lifespans compared to non-altered mice.
You know, it is deeply uncomfortable to hear about animal suffering.
It is, it's tough.
But from a scientific standpoint, having a mouse that accurately mirrors the human condition, that has the same seizures, the same deficits, I mean, that is the only way you can ethically test if an experimental drug works before giving it to a child.
The model is an absolute necessity. And JX didn't stop at a whole-body knockout. They also created a conditional knockout mouse.
How is that different?
They used a specific genetic tool called the CRISPR system.
CRISPR.
Okay? Think of LOX-T sites as microscopic bookends. Scientists use CRISPR to place these bookends on either side of the PRA gene in the mouse's DNA.
Okay, so the gene is bracketed.
Right. By themselves, the bookends do nothing. The gene still functions normally. But then you introduce an enzyme called Cre. Cre is a highly specific pair of scissors that only cuts the DNA located exactly between 2 LOX-P bookends.
That's a lock-and-key mechanism.
Exactly. And the brilliance of this is that scientists can control exactly where and when the Cre enzyme is produced.
Wait, really?
Yeah. They can engineer a mouse where the Cre scissors are only produced in the liver, or only in the skeletal muscle, or only in the brainstem. So instead of the mouse being born with the gene missing in every cell of its body, the gene is only knocked out in one specific organ.
Why do you need to do that?
It allows researchers to dissect the specific causes of different symptoms.
Oh, see.
If they knock out PURA only in the skeletal muscle and the mouse develops severe hypotonia but no seizures, they know the hypotonia is caused by a local muscle deficit, not a signal failure from the brain. It isolates the variables.
That is just incredible science.
Okay.
But the roadmap doesn't just rely on mice, though. It calls for human-induced pluripotent stem cells, or iPSCs, fruit flies, worms. Let's talk about the iPSCs first, because this sounds like pure science fiction.
It really is one of the most incredible technologies of the 21st century. Scientists can take a small, non-invasive sample of a patient's skin cells, fibroblasts, or blood cells.
Right.
In the lab, they expose those adult cells to a specific cocktail of proteins that forces them to forget their identity.
Forget their identity.
Yeah, they reverse engineer them back into stem cells. These are the induced pluripotent stem cells. Pluripotent means they have the potential to become almost any cell type in the body.
Wow.
Then, scientists change the chemical bath, coaxing those stem cells to grow into specific types of cells, like neurons or skeletal muscle cells, right there in a petri dish.
So you literally have a living model of a specific child's brain cells, carrying their exact, unique PURA mutation growing in a dish.
You do. You can study how their specific neurons fire, how their synapses form, and you can test highly experimental drugs directly on their living cells without ever putting the child at risk.
I have to challenge this, though.
Okay, challenge away.
A human brain is a wildly complex three-dimensional organ, right? With billions of connections, blood vessels, glial cells, a whole chemical environment. How does a flat 2D layer of cells in a petri dish accurately represent a 3D brain experiencing a seizure?
That is an excellent critical question, and it is the exact limitation of iPSCs. A petri dish is not a brain. You will never see a seizure in a dish, because a seizure is a network-level event.
Right.
What iPSCs give you is a cellular phenotype. You aren't looking for a seizure. You are looking at the individual neuron. Does it produce too much electrical activity? Do the dendrites, the branches that connect to other neurons, fail to grow long enough? Does the cell fail to clear away toxic waste products?
Oh, so you're zooming way, way in.
Right. iPSCs allow us to see the microscopic foundation of the disease. But you are entirely correct that to see if a drug stops a network-level event like a seizure, you eventually have to move back to the mouse model.
That makes sense. The dish shows you the broken brick, the mouse shows you the falling building.
That is a fantastic way to put it.
And then we have the fruit flies and the worms, Drosophila melanogaster and Canner habditis elegans. Why on earth do we need microscopic worms if we have human brain cells in a dish and mice in a cage?
It comes down to speed and scale.
Speed and scale.
Mice have complex brains, which is necessary to study complex symptoms. But mice take months to breed. They are incredibly expensive to house and care for, and you can only test a handful of drugs on a few dozen mice at a time.
Okay, yeah.
Worms and flies, on the other hand, breed exponentially in a matter of days. You can fit thousands of them into tiny, individual wells on a plastic plate.
They're built for high-throughput screening.
Exactly. You can use automated robotics to drop thousands of different, already FDA-approved drugs into thousands of wells containing PRE-mutated worms.
Thousands at once.
Within days, you can use cameras and AI to rapidly see if any of those thousands of drugs cause the worms to move better or live longer. It is a brute force numbers game that is impossible to play with mice.
Okay, but if I am a parent listening to this, and we are talking about making fruit flies and studying stem cells, aren't we taking a massive step backward into basic biology? I don't want a worm model. I want a treatment for my kid today.
That frustration is completely valid. It feels like moving away from the patient. But you cannot fix a complex engine if you don't know what the broken part actually does and how it interacts with all the other parts.
Yeah, that's true.
If you try to blindly fix it, you might cause a catastrophic failure. The basic science is the foundation, but, and this is the most crucial point of the roadmap, we are not just waiting on basic science.
We're not.
No. These models are not being built just to sit on a shelf and generate academic papers. They are actively being used right now to test the therapies of tomorrow. The basic science track and the therapeutic track are running in parallel.
Okay, so let's look at those therapies. The PURA Roadmap outlines 3 main avenues: antisense oligonucleotides, ASOs drug repurposing, and gene replacement therapy.
Yes.
Let's start with ASOs. I know they involve RNA, but what are they actually doing inside the cell?
Antisense oligonucleotides, or ASOs, are small, synthetic sequences of nucleic acids. Think of them as custom-designed pieces of genetic Velcro.
Genetic Velcro.
They are designed to enter the cell and bind to very specific molecules of messenger RNA, mRNA.
Let's bring back our factory metaphor. The DNA in the nucleus is the master blueprint stored in the manager's office. The mRNA is the photocopy of that blueprint that gets sent out to the factory floor so the workers know what to build.
That's right. Depending on what the basic science tells us about a specific PURA mutation, an ASO could be designed to do one of 2 things.
Okay, what's the first?
First, let's look at that dominant negative scenario where the mutated gene is producing a toxic saboteur protein. We can design an ASO whose sequence perfectly matches the mutated mRNA photocopy.
Just the mutated one.
Right. The ASO enters the cell, finds that specific bad photocopy as it's traveling to the factory floor, and binds to it.
It Velcros on.
When the ASO binds to the mRNA, it acts like a red flag. It signals the cell's natural waste disposal system to come over and shred that photocopy before the toxic protein can ever be built.
Oh, wow.
It intercepts the blueprint before it reaches the construction site. It knocks down the bad copy.
That is brilliant. It acts as a molecular spell checker, just deleting the typo. It stops the saboteur before it even exists. But what if it's haploinsufficiency? What if we just don't have enough protein?
Then you design a different ASO. This one targets the mRNA photocopy produced by the healthy copy of the PURA gene.
The healthy one. Why?
Instead of flagging it for destruction, this ASO binds to specific regulatory regions on the mRNA and essentially acts as a volume dial.
A volume dial.
It stabilizes the photocopy, allowing the factory workers to read it over and over and over again, upregulating the production of the healthy Pura Alpha protein to compensate for the broken copy.
So it's either deleting the bad sentence or turning up the volume on the correct sentence.
Precisely.
The second therapeutic track is drug repurposing. And this ties directly back to those thousands of worms and flies we talked about.
Yes, it does. Developing a brand new chemical drug from scratch takes 10 to 15 years and billions of dollars. It has to go through endless safety and toxicity testing.
Yeah, we've all heard how slow the FDA can be.
But if you can find a drug that is already sitting on a pharmacy shelf, already proven safe in humans, that happens to also alleviate PURA symptoms. You bypass a decade of safety trials. You can jump straight to testing efficacy.
The roadmap notes that a drug called Pyridostigmine is being looked at. Now, this is an acetylcholinesterase inhibitor usually used for a neuromuscular disorder called myasthenia gravis.
Right.
How does a drug for myasthenia gravis help a child with PURA syndrome?
It comes down to mechanism. Pyridostigmine works at the neuromuscular junction, the exact gap where a nerve ending meets a muscle fiber.
Okay, the gap.
When a nerve wants a muscle to contract, it releases a chemical messenger called acetylcholine into that gap. The muscle receives the message and contracts. Normally, an enzyme quickly sweeps in and cleans up the acetylcholine so the muscle can relax again.
Right, so you're not constantly flexing.
Exactly. Pyridostigmine blocks that cleanup enzyme. It leaves the acetylcholine in the gap longer, amplifying the signal from the nerve to the muscle. So for a PRA patient struggling with severe hypotonia, where the muscle tone is incredibly low, amplifying that nerve-to-muscle signal can improve core strength, head control, and swallowing.
That's amazing.
Drug repurposing is looking for these quick wins for symptom relief while the genetic cures are being built.
And the ultimate genetic cure is track 3, gene replacement therapy. This is the pursuit of a root cause fix.
The concept is elegant in its simplicity. You take a healthy, fully functional copy of the PRA gene and you deliver it directly into the patient's cells to replace the missing function.
But delivering it is the hard part, right? I mean, you can't just swallow a pill full of DNA. It will be destroyed in the stomach.
Right, you need a delivery vehicle. In gene therapy, scientists typically use a virus.
A virus.
Viruses have evolved over millions of years to be the perfect delivery vehicles. Their entire biological purpose is to inject their genetic material into host cells.
Oh, that makes sense.
Scientists take a harmless virus, usually an adeno-associated virus, specifically a variant called AAV9, and they completely hollow it out. They remove all the viral DNA so it cannot replicate or cause illness. Then they pack the hollow viral shell with the healthy PURA gene.
So AAV9 is just a microscopic biological delivery truck dropping off a healthy instruction manual.
Exactly. And AAV9 is chosen specifically because it is very good at crossing the blood-brain barrier.
Which is notoriously difficult to cross.
Yes. The blood-brain barrier is a tightly packed layer of endothelial cells that protects the brain from toxins in the bloodstream, but it also blocks most drugs. AAV9 can slip through and deliver the gene directly to the neurons.
But it requires immense precision. Right now, JAX and JAX Tomorrow are conducting gene overexpression studies.
They need to know the upper safety limit. What happens if they add too much Pura Alpha protein?
Oh, because if you're putting a new gene in, you might make too much of the protein.
Right. If we deliver a healthy gene, but the cellular factory goes into overdrive and produces an excess of the protein, is that toxic? Does it cause new neurological problems? You have to establish the exact therapeutic window, the safe dosage, before you ever attempt to inject millions of these viral vectors into a human child.
And while the scientists are working on engineering the delivery trucks, the regulatory agencies have actually been changing the speed limits on the highway.
They have.
In February 2026, the FDA issued updated draft guidance for individualized ultra-rare disease drugs. This feels like a tectonic shift in how we approach regulation.
It is a massive game changer. Historically, the regulatory system was built for drugs treating millions of people, statins for cholesterol or blood pressure medication.
Right.
To get a drug approved, you needed phase 3 clinical trials. That means taking hundreds, sometimes thousands of patients, splitting them into 2 groups, giving half the real drug and half a placebo, in a double-blind study where neither the patient nor the doctor knows who got what.
Which is mathematically impossible for PURA syndrome.
Completely impossible. When you have a global population of 800 people, scattered across 60 countries, and many are children with incredibly fragile, complex medical states, you simply cannot recruit enough people to run a statistically significant double-blind placebo trial.
So the system was basically rigged against rare diseases.
The old regulatory framework essentially locked ultra-rare diseases out of the system. A biotech company could have a cure sitting in a vial, but they couldn't afford the impossible trial design to get it approved. But this new draft guidance from February 2026 shifts the paradigm. The FDA states that for ultra-rare diseases, they will consider approval based on a plausible mechanism of action.
Plausible mechanism. What does that actually mean in practical terms for a researcher?
It means the burden of proof shifts from massive human trials to rigorous laboratory models. If researchers can definitively prove the genetic mechanism in the lab, if they can show in the JAX mouse model and in the human iPSC cell lines that the AAV9 gene therapy directly corrects the genetic defect, restores protein levels, and significantly improves the physical symptoms without severe toxicity, the FDA may offer a much faster, viable path to clinical approval.
Oh.
It acknowledges that the extreme precision of modern genetic medicine requires a modernization of the regulatory approval process.
It is thrilling. It is genuinely thrilling to think about AV9 delivery trucks crossing the blood-brain barrier and molecular spell checkers intercepting toxic blueprints, and a modernized FDA that actually understands the realities of ultra-rare disease.
It's a huge step forward.
But, and I want to pivot here, if I am a parent listening to this right now, doing the math on how many hours of sleep I got last night, making breakfast for my child, mixing their exact ratio of anti-seizure medications, setting up their G-tube feed pump, how do I advocate for my child's practical needs today?
Right.
Well, the scientists are doing their thing in the lab. What am I supposed to be doing with the local school board or the insurance company?
That is the daily marathon. Science provides the hope for tomorrow, but advocacy provides the survival for today. And navigating education, therapy, and bureaucracy requires an immense amount of strategic, exhausting thinking. Because there is no root cause cure available today, management remains entirely supportive and symptom-directed.
Which means treating the symptoms as they arise aggressively.
Yes. For example, let's look at speech and communication. A defining hallmark of PURA syndrome is a profound gap between receptive intelligence, what the child understands about the world around them, and expressive ability, what they can physically articulate.
So they understand everything, they just can't say it.
Almost all individuals with PURA remain nonverbal. Therefore, speech therapy focusing heavily on augmentative and alternative communication, or AAC, is not an optional extra. It is a fundamental human right.
A fundamental right.
You have to bridge that gap to prevent the profound frustration, anxiety, and potential self-injury that stems from being locked inside a mind that perfectly understands the world but cannot speak to it.
And then there's the constant, terrifying reality of seizures.
Seizure control requires relentless vigilance. Standard anti-epileptic drugs are the first line of defense, but PURA-related epilepsy is notoriously refractory, meaning it often stubbornly resists standard medications.
It just won't stop.
Right. Case reports and recent neurological reviews are showing that the ketogenic diet is showing remarkable promise in some of these drug-resistant cases.
Okay, wait, a diet. How does eating a high-fat diet stop an electrical storm in the brain?
The brain normally runs on glucose, derived from carbohydrates. When you drastically restrict carbohydrates on a ketogenic diet, the body's glucose levels drop.
Okay.
To survive, the liver starts breaking down fat into molecules called ketones, which cross the blood-brain barrier and serve as an alternative fuel source for the brain.
So you're changing the fuel.
But burning ketones changes the cellular environment. It alters the production of ATP, the energy currency of the cell, and it shifts the balance of neurotransmitters.
How so?
It often increases the production of GABA, which is the brain's primary calming, inhibitory neurotransmitter, and decreases glutamate, which is the excitatory neurotransmitter.
So you're calming the brain down chemically.
Exactly. By shifting the brain's fuel source, you literally stabilize the electrical activity of the neurons, raising the threshold required to trigger a seizure.
It's fascinating biology. But implementing a strict ketogenic diet, securing expensive AAC devices, and securing specialized physical therapies requires the school system or the medical insurance to actually pay for them.
Yes, that's the hard part.
And this brings us to the battlefield of educational and financial advocacy. We are going to use the UK context here as a specific global example, because the crushing bureaucratic hurdles are remarkably similar, whether you are dealing with a local council in London, a school district in Sydney, or an insurance provider in Chicago. In the UK, a child's support revolves around the EHC plan, the Education, Health and Care Plan.
The EHC plan is a legally binding document. It outlines a child's specific special educational needs and the exact provisions the local government is required to provide to meet those needs. Managing the annual review of this plan is one of the most critical, high-stakes advocacy tasks a parent undertakes.
Because you can't just wing it.
You cannot just show up to the meeting and hope the system has your child's best interests at heart.
The documentation you provided outlines a very specific battle plan for these annual reviews. It demands that parents treat the bureaucracy with the same empirical, ruthless precision that the scientists treat the genome.
You have to gather evidence meticulously. You become a data analyst for your own child.
A data analyst.
You keep diaries. A diary of attendance, a diary tracking spikes in anxiety levels, a diary tracking exactly when the child vocalized a new sound using their AAC device. If you want the local authority to provide specialized teaching or fund a dedicated one-on-one aid or purchase a specific piece of standing equipment, you must prove the necessity with undeniable data.
The guidance specifically warns against allowing vague language in these plans. It says to strike down phrases like access to. Why is that so dangerous?
Because access to a speech therapist is a legally unenforceable, meaningless phrase.
Really?
It could mean the child is sitting in a school building where a speech therapist happens to work in an office down the hall. It does not guarantee the child receives a single minute of therapy.
Oh, that is sneaky.
A strong EHC plan must be airtight, detailed, and specific. It must state the child will receive 45 minutes of direct one-on-one intervention with a qualified speech and language therapist twice a week, focusing specifically on AAC navigation.
That is very specific.
That is a quantifiable, legally binding provision. If the local authority fails to provide it, the parent has a clear, documented legal basis to appeal to the Sen Tribunal and force compliance.
And parents have to request all the professional reports from the occupational therapist, the educational psychologist, the school psycho, at least 2 weeks before the meeting.
Yes.
You cannot let them hand you a negative report as you walk into the room and expect you to process it on the fly.
You must have time to review the data, find the inaccuracies, formulate your counter-arguments, and submit your own written views in advance to set the agenda of the meeting.
It is mind-boggling. It's like you have to be a medical researcher to understand the molecular genetics of haploinsufficiency, a legal advocate to enforce the minutiae of the EHC plan, and a financial auditor all at once.
It is incredibly demanding.
Speaking of finances, the financial strain placed on rare disease families by society is staggering.
Yeah.
I mean, caring for a child with PURA is a $247 full-time job.
Yeah.
But families still have mortgages, they still have grocery bills, they still have to survive under capitalism.
And the state support systems designed to help are notoriously complex, often creating financial cliffs that trap parents. Looking again at the UK context as a prime example of this systemic friction, there is the carer's allowance.
Right, the carer's allowance.
To be eligible, a person must spend at least 35 hours a week caring for someone who receives a qualifying disability benefit.
35 hours is literally a full-time job.
It is. But the financial rules around claiming that allowance while trying to maintain outside employment to keep your career alive are exceptionally strict. The earnings cap is currently 196 SEK a week.
Wait, what? 196 SEK a week?
Yes. That is after tax, national insurance, and allowable expenses. If you earn 197 a week, you lose the entire allowance. The system creates a hard cliff.
Imagine being a mother, trying to calculate if picking up one extra Tuesday shift at work to pay for a broken washing machine will push her over the 196 SEK limit, causing her to lose her entire week's state support.
It is an awful position to be in.
It forces a parent to heavily restrict their working hours, severely capping their earning potential and destroying their long-term career trajectory.
It does. Now, the bureaucracy allows for specific deductions when calculating that 196 NARM limit, but you have to know how to play the game.
It's all a game to them.
For instance, you can deduct 50% of your pension contributions as an allowable expense, lowering your calculated income. Or if you pay someone else to care for your disabled child while you are working, you can treat those care costs up to 50% of your earnings as an expense.
Oh, that's something.
But there are caveats. The person providing the care cannot be a close relative like your spouse, your parent, or the child's sibling. It requires a meticulous, exhausting auditing of your weekly finances just to maintain a baseline level of state support.
If a parent is listening to this and they are feeling completely overwhelmed walking into one of these annual reviews or staring down a spreadsheet trying to calculate carer's allowance deductions, what is the single most important piece of leverage they have?
The parent's intimate daily knowledge of the child is the leverage. It is the most powerful weapon they have.
More than the doctors?
Yes. The professionals, the school psychologists, the local authority administrators, they might know the child for an hour a week or strictly as a file on a desk. The parent knows the child 168 hours a week.
Wow.
They know the exact precursor signs of a seizure. They know exactly how long the child can focus before fatigue sets in. That daily lived experience, when documented and presented confidently, is the most powerful evidence in any administrative room. But carrying that heavy administrative, financial, and emotional load requires immense personal resilience.
Which brings us to the core of everything, because if the scientific research we talked about is a vehicle driving toward a cure, And the EHC plans and advocacy are the steering wheel navigating the obstacles. Caregiver mental health is the engine oil.
I like that.
Without it, the friction becomes too great, and the entire machine grinds to a devastating halt.
It is a lifelong marathon. Caregiver self-care is frequently framed as a luxury, like a bubble bath or an hour away. It is not a luxury. It is the fundamental, non-negotiable fuel required to run a marathon of this magnitude.
It's essential maintenance.
Recognizing this, the PURA Syndrome Foundation partnered with an expert named Sharu Izadi to create specific, tailored self-care resources and workbooks for the PURA caregiver community.
I was actually reading through some of the quotes from parents in that workbook, and they are incredibly raw and powerful. One parent wrote, "You've got this, you deserve this." Another wrote, "Take things one day at a time, 1 hour at a time if the days are too heavy."
That is so true.
And then there was this piece of advice, which I think is so profound for any rare disease family. Do not Google. Find a group. Make sure you have a good team of doctors. Why do not Google? Isn't information power? Why tell a parent not to seek out information?
Context is everything. When PURA was first discovered in 2014, and for several years after, if a newly diagnosed parent typed PURA syndrome into a search engine, what did they find?
Probably just horror stories.
They found terrifying, sterile clinical jargon. They found worst-case scenarios documented in isolated medical journals. They found lists of severe deficits, profound delay, intractable seizures, brain abnormalities without any context of the joy, the personality, the resilience, or the lived reality of the child.
So it was just a laundry list of problems.
Google offered pure isolation disguised as medical information. It stripped the humanity entirely from the diagnosis.
But that is changing now.
It is changing because this community is actively, aggressively rewriting the internet. They are taking back the narrative. Now, when you search for PURA syndrome, you find the foundation, you find parent blogs.
You find real people.
You find stories of incredible resilience. You find actionable, hopeful science about CRISPR mice and ASOs. You find the hashtag #PuraPerfect campaign.
Which brings us to the immediate future, because we are looking forward. There were two major events on the horizon that perfectly embody this shift from isolation to mobilization. First, Rare Disease Day on February 28, 2026.
Rare Disease Day is a global movement. The PURA Foundation is asking families to flood social media with their hashtag #PuraPerfect stories. They use zebra graphics.
Oh, the zebra. Explain the zebra.
In medical school, doctors are famously taught, when you hear hoofbeats, think horses, not zebras meaning. Assume the most common diagnosis.
Right, it's probably just a cold.
Exactly. But rare disease patients are the zebras. It is a day to educate local schools, to lobby state legislatures for better funding, and to visually, undeniably demonstrate the strength and numbers of the community.
And then the massive culmination of all of this connection, the in-person PURA Syndrome Conference in Munich, Germany, from June 26th to the 28th, 2026.
The foundation has been organizing these global conferences to bring the scientists, the clinicians, and the families into the exact same physical space. The Munich conference will feature in-depth sessions on the latest PURA Roadmap research, parent discussion groups, and vital, unstructured social time.
As we look toward an event like the Munich conference, what actually happens when these parents, who might be the only family in their entire city or even their entire country dealing with this, what happens when they finally get into a physical room together?
The isolation shatters. When you walk into a room and you see 50 other children who move like your child, who use AAC devices to communicate like your child, who have the same unique, beautiful physical traits as your child, you realize you are part of a powerful, unified front.
I'm getting chills just thinking about it.
They can talk to the genetic researcher from the Jackson lab or the clinical informatics expert from Southampton over a cup of coffee in the hallway. The imposing gap between the sterile laboratory bench and the chaotic family living room completely disappears.
We have covered an immense amount of ground today, from the intricate biology of the Pura Alpha protein regulating mRNA and the creation of CRISPR knockout mice, to the brutal realities of navigating EHC plans and the financial cliffs of caregiving. We've talked about the past 10 years of discovery and the incredibly bright, aggressive pipeline of research moving toward the 2026 horizon.
It is a vast amount of information to process. But if there is one overarching, undeniable theme from all these sources, it is that the pieces are moving. The scientific models are built and breeding.
They are.
The regulatory pathways at the FDA are shifting in our favor. The global registries are active in collecting data. The ecosystem is primed for a breakthrough.
Which brings us to the end of our 31-day journey. To you, the listener, I want to say thank you. Thank you for letting us into your ears, your cars, your kitchens, and your quiet moments over this past month. We acknowledge the profound exhaustion. We acknowledge the fear that inherently comes with a rare genetic diagnosis.
Hmm.
But most importantly, we acknowledge the profound, relentless, world-moving love that drives you and drives this entire community forward.
The progress we discussed today, the mice, the biobanks, the ASOs, none of it is happening by accident. It is happening because of the sheer force of parental love demanding answers from the universe.
I want to leave you with one final thought, a thought to mull over long after this audio stops playing. We talked today about how PURA syndrome is almost always a de novo mutation. In the strict, cold, clinical language of genetics, that means it starts fresh with your child. It was not handed down to you through generations of your family tree. You did not inherit it. But I want you to step back and think about what you are building right now, today. By logging onto that portal late at night and donating your child's data to the global registry at Southampton. By participating in the biobank. By fighting tooth and nail armed with data for that AAC device in a hostile school board meeting. By sharing your hashtag #PureAPerfect stories on Rare Disease Day. And by showing up with empathy for another terrified parent in a Facebook group.
It matters.
You are completely redefining what the word inherited means in the rare disease world. You are actively building a new kind of inheritance. Because of the exhausting, relentless work you are doing right now, the next parent who receives this terrifying diagnosis in 2027 or 2030 will not inherit a blank page. They will not inherit a terrifying, isolated Google search filled with clinical jargon. They will inherit a map. They will inherit a fully characterized CRISPR mouse model, a clinical pipeline full of ASOs and gene therapies, a modernized FDA pathway that understands their struggle, and an absolute army of friends waiting to catch them. You aren't just caring for your child today, you are creating the ancestral legacy of hope for every Pura family of tomorrow. Thank you for joining us on the Deep Dive.